This rendering captures the exact molecular interface where immune memory is forged. We are looking at the precise atomic docking between an antibody, a pathogenic peptide, and a cellular display. Zooming out from this micro-interaction to view the complete complex and the cell membrane reveals the broader architecture. The adaptive immune system functions as a highly precise, engineered network, operating with strict mechanical logic. To understand how this network functions, we have to perform a structural analysis of the two master biomolecules that make it possible, immunoglobulins and the major histocompatibility complex. Mastering the physical architecture of these two molecules is the only way to map how the body accurately distinguishes its own tissue from a foreign invader, preventing total biological collapse. This graphic illustrates the dual hardware model that secures the host. The system relies on two completely different recognition technologies operating in parallel. The first pillar consists of the immunoglobulins. These are soluble sentinels. Their mission is to sweep through extracellular fluids, physically hunting down and neutralizing free-floating threats. The second pillar, the major histocompatibility complex, or MHC, operates on the cellular level. These are permanent cellular displays, anchored directly to the cell membrane, serving as diagnostic windows for T cells to scan. By tearing down the structural blueprints of these two distinct pillars, we can map the complete trajectory of an immune response, from the first atomic contact all the way to clinical pharmacology. Let's begin with the baseline structural unit of all antibodies, shown here, the modular Y blueprint. The chassis is built from 4 distinct protein chains. 2 long, heavy inner chains form the main stem and arms, flanked by 2 shorter light chains on the outer edges. Covalent disulfide bonds secure these chains into a single, stable unit. Functionally, the molecule is split into 2 regions. The constant region at the base dictates how the wider immune system will respond, while the variable region at the tips determines the antibody's binding specificity. At the extreme tips of these arms lie the hypervariable regions. These small domains provide the exact atomic interface required for unique complementary binding to an antigen. This physical modularity allows the antibody to maintain a stable, uniform core structure, while continuously swapping out the extreme tips to bind virtually any pathogen in existence. This diagram maps how structural variations on that basic Y blueprint scale up to create an entire arsenal. The variations occur across 3 specific tiers: isotype, allotype, and idiotype. The idiotype defines the millions of unique binding specificities at the tips of the antibody. The isotype defines the structural class of the heavy chains, which determines the antibody's ultimate biological mission. This matrix details the 5 major isotypes at immunoglobulins. Notice how their distinct geometric shapes dictate their specific roles in the body. IgG is the abundant secondary responder, small enough to cross the placenta. IgA is built for mucosal immunity, protecting the respiratory and gastrointestinal tracts from pathogen attachment. IgE binds tightly to mast cells to mediate parasitic defense and allergic reactions, while IgD functions primarily as a surface receptor for initial B-cell activation. Then there is the extreme geometry of IgM. Its massive pentameric structure provides supreme physical avidity, making it the ideal first responder to aggressively trap and clump pathogens together. The large-scale physical geometry of an antibody dictates its movement through the body and the force of the resulting immune response. While antibodies sweep the fluids, T cells rely on a physical cellular interface to scan for threats. This is the major histocompatibility complex. The system uses 2 primary sentinels. MHC Class I acts as a general monitor, present on nearly all nucleated cells in the body. MHC Class II is restricted to professional antigen-presenting cells, such as macrophages and dendritic cells. Looking closely at these 3D models, the primary mechanical difference between the 2 classes lies at the very top of the structure, in the groove architecture. Class I features a closed topology, physically restricting it to holding short, tightly contained peptides. Class II features open ends, allowing it to accommodate much longer, draping peptide chains. The physical capacity of these grooves acts as a rigid mechanical filter. It ensures that cytotoxic and helper T cells only engage with the specific size and type of pathogenic data they are biologically programmed to handle. This brings up a strict logistical problem. How do specific pathogenic fragments find their way into these exact surface grooves? This flowchart details the solution, distinct molecular routing. We'll start with the endogenous or intracellular pathway. Internal threats, like viral proteins, are mechanically shredded by a complex called the proteasome. These shredded fragments are transported via Tap proteins directly into the endoplasmic reticulum, integrated into newly synthesized MHC Class I molecules, and driven to the surface for inspection by CD8 positive T cells. The exogenous pathway operates on external threats. Antigen-presenting cells engulf free-floating bacteria via phagocytosis and degrade them in highly acidic endosomes. To prevent premature binding during synthesis, MHC Class II is built with a temporary invariant chain blocking its groove. This block is only removed and replaced with the exogenous peptide right before presentation to a CD4 positive helper T cell. MHC displays function as the highly regulated end products of two completely distinct intracellular manufacturing and sorting pipelines. This diagram brings both structural pillars together into a master synthesis. Adaptive immunity achieves dominance by coupling MHC presentation and immunoglobulin production into a continuous, self-amplifying loop. It begins with a professional antigen-presenting cell swallowing a threat. It routes the exogenous peptide to its surface using MHC Class II and uses that physical display to activate a helper T cell. That activated T cell acts as a bridge. It locates a specific B cell and triggers its maturation into a plasma factory, rapidly accelerating the production of specific immunoglobulins. The cycle completes as new antibodies flood the system, coat the remaining bacteria, and mechanically drag them directly back to the presenting cells for destruction. Our entire modern framework for clinical immunology relies on understanding this exact biomolecular architecture, as shown in this four-quadrant matrix. We exploit immunoglobulin structures for diagnostics, using specific ratios of IgM and IgG in ELISA testing to determine the stage of an infection, and we engineer monoclonal antibodies for highly targeted therapeutics. Conversely, the highly polymorphic genetics of the MHC structure dictate the strict rules of organ transplants, requiring precise HLA typing to prevent graft rejection. These same structural variations map directly to predispositions for specific autoimmune diseases. Let us return once more to the physical point of contact. In the immune system, structure strictly dictates function. Total mastery of this physical architecture is the foundation for modern clinical intervention and the ultimate mechanism for saving human lives.